Hi students, I welcome you for this session to know more about the immune system related to the tumor immunology. What is actually a tumor? Now the study of tumor or cancer is called oncology. The study of tumor or cancer is called oncology. So cancer, the word is derived from carcinose. It is derived from carcinose. Cancer is derived from carcinose, the Greek word referring to crab. Now the crab is always, if you are observing the crab is sitting idle. There is no movement. But if you are observing carefully near the mouth, there are some activities are going on by the movement of the legs or by movement of the appendages, we can say, in the mouth. Just like that, we cannot see the cancer outside. Something is going on inside without any external expression. After just reaching the maximum growth, only we have the external symptoms. That's why it's called as cancer. Carcinose, the word referring to, that is what is called the crab. The study of cancer tumor is called oncology. In some cases, cancer is also caused by the genes. Such genes which cause cancer are called oncogenes. Such genes are called oncogenes. The genes which cause cancer are called oncogenes. Now let's see, what is tumor? So tumor is otherwise called as neoplasm. The other name for tumor is neoplasm. So it is nothing but a group of cells whose growth has gone unchecked. That means the abnormal uncontrolled growth of cells occur in a tumor. That is called a cancer, tumor, and neoplasm. Abnormal uncontrolled growth of cells, that is called tumor and neoplasm. That's why I said it's a group of cells whose growth has gone unchecked. They are going on and they're going on just normally dividing, resulting in growth. Then when these tumor cells continues to grow and invade healthy tissues, it is called cancer. It is called cancer. So when the tumor cells continue to grow and invade healthy tissues, it is called cancer. Now, in some cases, the cancer cells are spreading to different parts of the body from the tumor of origin. So from the place of origin, the tumor cells are spreading to other parts of the body through the bloodstream or the limb. In such cases, it is called secondary cancer or secondary tumor. Otherwise called what is known as metastasis. What is metastasis? The cancer cells spread from tumor to other parts of the body and produce what we have the secondary growth of cancer. Such a secondary growth of cancer is called metastasis. So the secondary growth of cancer is always away from the primary cancerous tissue of origin. So that's about metastasis. Based on the characteristics, we can classify the tumor into two different types. Now let's see about that. Based on the characteristics, the tumors are classified into two types. One benign tumor, another one malignant tumor. Benign means harmless and malignant means harmful. Now what are the differences between these two? Here we have the cells are, though dividing rapidly, they are non-cancerous in nature, do not produce cancer, harmless, doesn't cause death of the individual. Here the non-cancerous tissues are growing indefinitely. The growth is there, cell division is rapid but harmless. And once they are growing, they are restricted or localized to a particular region. They do not invade other tissues. There is no secondary growth of cancer with reference to benign tumor. It doesn't produce secondary growth. Secondary growth means spreading to other parts of the body. So they do not invade other tissues. Generally speaking, this tumor is harmless. This tumor is harmless. Now what about the malignant tumor? Malignancy, harmful, highly fatal. Now here the cells are cancerous. The meaning for that one, they cause damage to the body. They undergo growth indefinitely by dividing and so on, but the cells are cancerous. The meaning for that one, they can spread to other parts of the body and they produce secondary growth of cancer and metastasis. So, they detach and migrate into the surrounding tissues. From the cell mass, the primary cancer, the cells get detached and transported to the surrounding tissues. They migrate into the surrounding tissues through the bloodstream or what we call the lymph. So they produce what we call the secondary growth. Here there is no secondary growth formation, there we have secondary growth formation. So this tumor is highly fatal or harmful. So another difference what I mentioned, there is no secondary growth. The meaning for that one, the cells do not invade the surrounding tissues. Where in the case of malignant tumor, we have secondary growth. The cells invade to the surrounding tissues by migration through the blood and the lymph stream. Okay, that's about the two types of tumor. Now let's see what are the properties of cancerous cells and what are the differences between, that is, the cancerous cells and also the normal cells. Okay, let's see. What are the differences between the normal cells and cancerous cells with reference to certain basic properties? Here I mentioned about two properties. We also have many properties. We have to go for the differences next. Now in normal cells, the growth and differentiation is highly controlled and regulated. The cells undergo growth, they are being differentiated in different types of tissues. They are either what is called the gene control or physiological control. The growth and then differentiation. Both are controlled mainly by the genes. There is a mechanism of controlling and regulation. But in the case of cancer cells, they are not under anybody's control. There is a breakdown of this regulatory mechanism. They are going on growing. There is no differentiation. All the cancer cells are more or less similar. Here the cells are differentiated into different types of cells to perform the functions, but the cells are growing and there is no differentiation, so there is no controlling mechanism. Now the second property of normal cells is contact inhibition. During cell division, new cells are formed. When they come in contact with one another, they prevent or inhibit further cell division. That means they inhibit uncontrolled cell division. They are allowing the cell division but prevent or inhibit the uncontrolled cell division. That is called contact inhibition. So the contact inhibition is a property of normal cells. The contact is a stimulant which prevents what is called further cell division or uncontrolled cell division in normal cells. But in the case of cancerous cells, they lost the property of contact inhibition. Though the cells are in contact with one another, during cell division, more and more cells are formed. Now the contact mechanism is not working. Though they contact with one another, they are going on dividing to form more and more cells. So this is called loss of contact inhibition. So a contact is a stimulus which inhibits cell division. That is a property of normal cells. But in the case of cancer cells, though they come in contact with one another, what will happen? They are going on dividing. The mechanism is not working. So they lost the power of what is called contact inhibition. As a result, the cancer cells are growing continuously and they produce a mass of cells. That mass of cells is called what is known as the tumor. The mass of cells resulted from continuous division of cancerous cells is called what is known as tumor. What are the differences between normal and cancerous cells? I am taking merely 8 characters. Here I represented 6 characters. 2 more characters to be explained after this. Now about the nature of the nucleus. So in the case of normal cells, the nucleus is small, uniformly shaped. There is no just a differentiation in the shape of the nucleus. All nuclei are same. That is in the case of normal cells where they have a group of cells. Now in the case of cancer cells, the nucleus is large. It is variable in shape between the cancer cells. It's variable in shape between the cancer cells. Then about the amount of cytoplasm. The amount of cytoplasm is relatively large. The volume of cytoplasm is relatively large. We see that one, this is a cytoplasm, more cytoplasm. But here relatively small in volume. The cytoplasm is relatively small in volume. As the nucleus is large, we have only small amount of cytoplasm. Now what about the size and shape? Conformity in size and shape. Conformity in size and shape means definite in size and shape. But in the case of cancer cells, the nucleus, the size and shape of the cell is, the size and shape of the cell, not nucleus, the size and shape of the cell is not uniform. The cells are variable in size and shape. So size and shape of cells here, not the nucleus, size and shape of the cells. So conformity in size and shape, that is uniform, no variations. But in the case of cancer cells, normally the cell size is variable, not uniform in size, not uniform in shape, different in size, different in shape. So the cells are normally, actually know that one, somehow not differentiated but different in size and shape. Now the next one, the arrangement of cells. So the cells are arranged together to form definite discrete tissues. But here the cells are not organized, that is called disorganized. The cells are disorganized. So there is disorganized arrangement of cells. There is no regularity here and there in a rough manner, not uniformly distributed, not organized well. So disorganized arrangement of cells. Now the cell structure. Now we possess differentiated cell structure. The cell structure is differentiated, having nucleus, cytoplasm, membrane, etc. So normal specialized feature is being lost. There is no definite actually structure for what is called the cancer cells. See here represented one cell having some membranous outgrowths. So here this is a normal cell, this is what is called actually a cell which lost its normal characters. Then about the cell markers. To identify the cells, each and every cell has some markers on the cell membrane. They are called cell markers. Here the cell markers are presented normally on the surface of the cells, uniformly normally. So normal presentation of cell surface markers. But here certain cell markers are elevated, certain cell markers are depressed. So we have elevated expression of certain markers. What we can say the cell surface markers. Some are elevated, some are just depressed, some are just having the same line on the surface of the cell membrane, etc. That's about the cell markers nature. And two more. The next one about the nature of rate of cell division. So the normal cells are dividing at a lower rate. So lower number actually the cells are dividing at a lower rate. But you see that one, the cancer cells are dividing at a faster rate, so we have large number of dividing cells. So lower level of dividing cells, large number of dividing cells. Then about the cellular boundaries. Here the normal cells are clearly demarcated. Demarcated means they have cellular boundaries. So the cellular tissues are clearly demarcated. That means definite boundaries we can find out. But in the case of cancer cells, poorly defined tumor boundaries. You cannot just find out the boundary easily. Here you can find out the boundary easily, but there in the case of cancer cells, you have poorly defined what is called cellular boundaries, what is called the tumor boundaries. No definite boundaries between the cells or between the tissues of the tumor. But here, definite cellular boundaries are there, so we have clearly demarcated boundaries. That's about the next 2 points. I'll write it down here after wrapping this one. As I mentioned earlier, the remaining 2 differences between the normal cells and cancer cells related to the rate of division and also cellular boundaries. In normal cells, you have lower levels of dividing cells. The rate of division is low. And in just cancer cells, because of rate of division is high, see higher number of dividing cells we can get. Now the cellular boundaries, the cellular tissues are clearly demarcated. We have cellular boundaries, you can identify the cellular boundaries. But in the case of cancer cells, poorly defined tumor boundaries. See that one closely arranged cells, we cannot find out the boundaries. But here the cells are loosely aggregated so that we can find out the boundaries, the cellular boundaries. So these are some of the differences between the normal cells and cancer cells. What is happening during malignant transformation? That is nothing but the conversion of normal cells into malignant cells. When the normal cells undergo malignant transformation, they acquire some surface antigens and also lose some normal antigens. What is surface antigen? The antigens present on the cell membranes of the malignant cells, which induce what is called the immune response. Now, to stimulate the cancer cells, the antigens are not coming from outside. They are present inside, that on the surface of the cancer cells. They act as antigens, hence called surface antigens. They stimulate the division. They stimulate the division of the cells. So the rate of division is increase and increase. This is because of the surface antigens present on the surface of the cell membranes. Now, these antigens, surface antigens only induce immune response. Normally, both cell-mediated immunity or cellular responses and humoral immunity, they are taking part what we have in the case of malignancy. Both the immune responses we could see, but they are unable to what we have control the cell division. Though we have the immunity of what is called actually cell-mediated and humoral. Both fails to what is called control the cell division, though they are involved. Now, the cancer cells normally can avoid immune detection. They cannot be detected by what is called the immune system. Why? Because the antigens are not coming from what is called outside. They are nothing but actually the processes performed by the abnormal cells. Here I can write, cancer cells can avoid immune detection. Why? As antigens are not foreign bodies, they are present only at the surface of the malignant cells itself, but abnormally functioning body cells. Abnormally functioning body cells are cancer cells having a surface antigen on them. So anyway, now cancer cells can avoid immune detection. They cannot be detected by the immune system. This is because immune system can detect only when there is a foreign substance and antigen enters to the body. But here the antigen is present on the surface of the cells, making the cells to function abnormally. That's why here I mentioned about. So that is the reason why we cannot treat the cancer cells easily. This is because of that one, cell surface antigens, abnormally functioning cells, but the antigens are not coming from outside. So this is about the malignant transformation, what is happening during that process. Acquiring surface antigens, then avoiding the immune justice system because they are not foreign bodies but abnormally functioning cells. That is the reason why the cancer cells cannot be easily treated. Now, what is the concept behind immunological surveillance? This actually looking after the immune system, how far it is working, what is the reason it is not working against cancer. Now, the concept of immunological surveillance mechanism we have, looking after the what is called the functions of immune system, postulates that the primary function of the immune system is to seek and destroy the malignant cells. Just to seek to find out which is the malignant cells and destroy it. And those malignant cells which arise by somatic mutation. So the primary function is to find out the malignant cells which arise by somatic mutation and destroy them. This is a mechanism. But this mechanism reduces. Why? Number one, as a result of aging, or number two, we have due to congenital or acquired immunodeficiencies. So the surveillance mechanism is being reduced as a result of that is aging, that's the number one factor, or due to congenital or acquired immunodeficiencies. As a result, the surveillance mechanism is lost or its function is reduced. Suppose if the surveillance mechanism is effective, cancer should not occur. If there is a development of cancer or tumor, it represents the lacking or lapse of what is called surveillance mechanism. If the surveillance mechanism is ablative, now there is no development of cancer. If it is lapse, then there is a development of tumor. So once the tumor is developed, it is because of the lapse of surveillance mechanism, immunological surveillance mechanism, one which is watching the immune system. So that's about immunological surveillance. Now we have to go for what is immunotherapy. How can you achieve it? What is immunotherapy? So it is therapy. Therapy means the treatment of cancer through what is called immunotherapy method. So immunotherapy of cancer. So it can be cured by means of immunotherapy method. Through immune system products, the treatment can be made to just actually to prevent or arrest cancer. So immunotherapy is also called as biological therapy. Now it uses substances made by the body or the substances made in a laboratory, particularly monoclonal antibodies. What are monoclonal antibodies? These antibodies contain a clone of antibodies which are all similar in function, structure and fighting against the same antigen. A clone of antibodies. A clone means similar. A clone of antibodies normally being made by means of a technology, hybridoma technology. We'll be studying later, I'll tell you. So monoclonal antibodies, similar antibodies which are attacking the same antigen. So what for? To resist or promote the functions of immune system. To resist the immune system against cancer or to promote the functions of immune system. Now what is the importance of immunotherapy? Immunotherapy. So after that one radiation or any other methods, if the malignant cells have been removed, what is called the residual cells, after the gross tumor has been removed. So immunotherapy appears to be important. What is for? In getting rid of the residual tumor cells, the residual what is called the malignant tissues after the gross means the large total tumor has been removed. So if you are doing the surgery, if the gross tumor has been removed, still we have some residues of malignant cells. These malignant cells can be removed with the help of immunotherapy. Okay, how can you get a better result of cancer treatment? So the better result of cancer treatment is to follow an integrated approach to therapy. Not one therapy, a combination of many therapies. That is called integrated approach to therapy, combining surgery, radiotherapy, chemotherapy, taken orally, and also immunotherapy. So you can just get a better result only by a combination of that is different approaches which are all integrated together, just like surgery, radiotherapy, chemotherapy and immunotherapy. That's about what we have the immunotherapy of cancer, how to treat cancer through immunotherapy method. Okay. Now the next one, what is the scope of immunology? A few just words only I want to tell you what are the different fields you can just get it through immunology. Either for what is called better employment in the hospitals, either in private or government hospitals or different fields of immunology. That's what is called the scope of immunology. Now the different fields of immunology which are grouped under one category, what is called the scope of immunology. Number one, immunotherapy, what is studied. Cellular immunology, immunology at the cellular level. Tumor immunology, that is about the cancer. Translational immunology, what is happening at the time of translation of proteins. Transplantation immunology, that is a grafting, organ transplantation. Then clinical immunology, the one which is being used in the clinical field. Then microbial immunology, how far the microorganisms actually being, that is normally affected by the immune system. Now allergy immunology, we have studied already the hypersensitivity related to immunology. Neuroinflammatory disorders, we have a number of disorders of the brain due to inflammation of the nerve cells. Then vaccine immunology, the relationship between immunology and the vaccines. Then inflammatory disorders, we see already the inflammatory reactions because of what we have, that is injury and infection, and that one can be studied because of what is called the effect of immune system. Ocular immunology, related to what we call the eyes and inflammation of the eyes. These are all the different fields which are being studied under the scope of immunology. With this I concluded the part immunology, that is health and diseases, and I have to go back to that is health and disease one part, alcoholism and drug abuse, somehow related to mental health also. This is thing to be completed under what we call the 7th chapter, human health and diseases. Alcoholism and drug abuse, their effect, how to prevent them.